Hello, everybody, and a warm welcome to this webinar on the significance of structural biology for Africa, the recent advances and future perspectives in vaccine research and development. This webinar is the fifth in a series of planned webinars that have the goal of providing information on available career options in the field of vaccinology. My name is Caroline Mate, the Vaccinology Workforce Development Program Planning Manager at the KEMRI-Wellcome Trust Research Program. And together with my colleague Marianne, we will be the facilitators and moderator for these sessions. Before we start, a few housekeeping matters. Kindly note that this webinar is being recorded and that please feel free to use the chat option to share your questions during the presentation, and we will address them during the Q&A session. We'll have a few open slots for people to directly ask questions to the presenter. And if your internet is stable, you can raise your hand and we'll be able to directly ask your question. As earlier mentioned, this is a series of planned webinars with the goal of providing information on available career options in the field of vaccinology. This is being run under the KEMRI-Wellcome Trust Research Program, which is a world-renowned health research unit of excellence based in Kilifi and Nairobi in Kenya, as well as Mbale in Uganda. Over the years, we have excelled in the use of novel ideas and collaborations to achieve better health for Africa, while also developing African scientific leaders. Our vision at the program is to undertake cutting-edge and novel research that is relevant to national, regional, and global needs. It is under this program that we have the Vaccinology Workforce Development Program that aims to expand the technical know-how and capability of sustainable end-to-end vaccine research and development in Africa through training and knowledge exchange activities in the vaccine ecosystem. This involves areas such as target product profiling, research and discovery, preclinical and clinical studies, biomanufacturing, regulatory affairs, and deployment. The workforce development is carried out through three channels. One, having internships at human vaccine manufacturing institutions that aim at establishing a mentorship scheme that links interns with senior vaccinologists and health or market systems, and engagement with academic institutions globally so that they can get to share and strengthen experiences in vaccine research and development and manufacture and also enhance career development. The second pathway is through a fully funded 2-week master's level course in vaccinology that is presented in multi-languages to be able to successfully, where successful applicants are selected based on their direct involvement with vaccine research and their excellence in professional and academic achievement. The last platform is through webinars such as this that target early career scientists, graduate and undergraduate university students in Africa and globally, and so that it helps to provide information on possible career options for this particular audience, so that it's able to help with informed career decision making. The program is in alignment with the Africa Union Partnership for Africa Vaccine Manufacture ambition to increase local vaccine production to meet 60% of Africa's routine immunization needs by the year 2040, and strengthen the capability for vaccine research, discovery, and manufacture in Africa. This webinar is part of that engagement. Today, we are glad to have Dr. Emmanuel Nji, who will be discussing the significance of structural biology for Africa, and he will be looking at what are the recent advances and future perspectives in the vaccine research and development space. So I will give just a brief bio of our speaker and then invite him to do his presentation. So Dr. Nji obtained his undergraduate degree in biochemistry in Cameroon, and then carried out a master's thesis that enabled him to enhance his skill in membrane protein structural and functional biology at Imperial College London, and secured funding for his PhD studies in Trinity College, Dublin. Dr. Nji has always retained a strong vision for supporting the training of African scientists. And while in Stockholm, he co-founded and still currently leads a nonprofit organization called Biostruct-Africa that brings together an international faculty of well-respected structural biologists who come together to organize workshops for African-based scientists to build capacity in structural biology. He is currently a Wellcome Trust International Intermediate Research Fellow, and his research is focused on implementing good laboratory practice to expound on the atomic structure and functions of membrane transport proteins that are relevant to animal, human, and plant diseases, while gaining insights into how these molecular machines work. And this aids in the rational design of new drugs and vaccines, as well as guiding in the optimization of already existing medications. We are happy to welcome you, Dr. Nji. Over to you for your presentation. Thank you.
Thank you very much, Caroline, for that generous introduction. Can you confirm, please, if you can see my slide?
Sorry, Doc, I think you're muted.
Oh.
Can you hear me?
Yes, I can hear you. I don't think I'm muted. Can you hear me? Hello? Can you hear me? Can you hear me?
Okay.
Can you hear me?
Hear you?
Hello, Caroline. Can you confirm that you can hear me, please?
Loud and clear, yes.
Okay, thank you so much. Sorry for that disturbance there. Yeah, I was saying thank you very much for the generous introduction. The title of my talk is The Significance of Structural Biology for Africa. And I'm going to be looking into how structural biology can be utilized for drug discovery and vaccine design. So my talk basically is making the case of why we should be actively conducting structural biology research in Africa. So you've already introduced me, but just to give you a brief introduction of myself, I was born in Cameroon. I did my undergraduate degree there. My research focused on the structural determination of drug target and to functionally characterize these drug targets. And I'm actively involved in capacity building for Africa-based researchers in the field of structural biology. And in addition, I'm actively involved in antibiotic resistance research to inform health policy on antimicrobial resistance. So my talk is going to be divided into 3 sections. So the first part is the significance of structural biology for Africa. So I'm going to highlight the evidence from the literature. And then in the latter part of the presentation, I'm going to be giving you evidence from Africa-based researchers. So we've basically conducted a survey from 100 Africa-based scientists on how structural biology is going to impact their research, and I'm going to be presenting that result. And then in between, I'm going to introduce to you the organization Biostruct-Africa, which I am CEO, our vision, and the recent progress we've made. So what is structural biology? Simply is to take pictures of protein molecules and nucleic acid, DNA and RNA. And why do we do that is to see their molecular details. And this helps us to understand their function, and we can utilize this information to design new drugs and vaccine to target a particular gene of interest. So the usual scenario is to be able to explore a biological space. So you identify a gene of interest to you, and then you determine the structure, utilize the structure to screen a chemical space to potentially identify a drug lead that can be developed further. In the case of vaccines, you definitely design the protein and then solve the structure, and that can help you in terms of providing information about your antigen and epitopes that you can utilize for your vaccine discovery. So in the first case, I'm going to explain to you how recently I have utilized structural biology to understand the mechanism of a drug target. And the protein that I'm looking at here is called, it's a lysine specific permease. So lysine is an amino acid. So this protein lys P here, it transports lysine from outside the membrane to inside the membrane. And what does it do? It's a unique transporter in the sense that it has transporter function, and it also has a regulatory role. This protein helps bacteria to survive under acidic stress condition. So when the environment is very acidic, such as in the mammalian stomach, the lysine transporter transport lysine and interacts also with another protein called CAT-C, which upregulates CAT-BA-operon. And this helps to decarboxylate the lysine to cadaverine, which is exported outside. It's an alkaline product which neutralizes the pH of the environment, which is essential for the bacteria to survive. So how does the protein transport lysine, and why does it transport only lysine? Why is it specific for lysine? So we did some functional assays. So we have an artificial membrane. So we insert the protein into that membrane and do transport assay. So it's a radioactivity transport assay. So if it transports the radioactive substrate, you can detect that. So using that assay, you can see that it's specific for lysine, and then it's inhibited by an analog of lysine, which is called L4-thiolysine. So basically, you can see here that it's a very specific lysine transporter. The other 19 amino acids were not able to compete for the transport of lysine. And as you can see here with binding studies, you can see the KD for lysine is much higher than even arginine, in which we didn't see any binding for arginine, which are two positively charged amino acids. So we utilize the protein and inject into llamas and raise nanobodies, which is some sort of an antibody. And in combination with the lys-P, improve the stability by 20 degrees. And as you can see the characterization here on size exclusion chromatogram. So in the presence of the complex, that's LISP and the nanobody, you can see a shift in the size exclusion chromatogram. And if you run this on an SDS page, you can see two species co-eluting together. And this is LISP alone, and this is the nanobody alone, which are controls. So secular dichroism spectroscopy of the purified free LISP shows predominantly alpha helical profile, and this gives us the indication that the protein is good enough for cryo-electron microscopic study, which is a method to determine the structure of a protein. So this is the grid which we use these are the particles on the grid, and these are the 2D classes, and this is the structure of the protein. So I'm going to go into detail at some point to tell you about the different methods of determining protein structures later on in the presentation. But the take-home message here is that you can utilize structural biology to understand the mechanism of a protein. So we've now solved the structure. It gives us the LISP nanobody complex. So the nanobody binds on the periplasmic side of LISP. So both the C and N terminus of the LISP is located in the cytoplasm, and LISP has 12 transmembrane segments. As you can see here, this is the lysine, and this also reveals some other features of the protein, which there's a beta hairpin and a lateral helix. As you can see, the nanobody here. So we've been able to identify the residues that are involved in the binding of lysine and the transmembrane helices that are coordinating the lysine. And also we docked tyrosinase into the electron density. We found this, it overlays with the lysine, indicating that the binding site is the right one. So based on that, we were able to propose a mechanism. So as you can see here, the lysine is sandwiched between tryptophan 105 on TM3 and phenylalanine 215 on TM1B. So basically, this tryptophan acts like a gate. To screen only for lysine, because it forms a strong cationic pi interaction with the lysine. So based on that, we propose a mechanism, which you can see here. This is the lysine. It goes into the structure. And as you can see here, you can see the tryptophan and the phenylalanine. So the lysine is sandwiched between the two, and it's going to be released into the cytosol. So based on that, we have a mechanism and we have a binding site, and this information can be utilized for screening inhibitors, which we are currently working at the moment. As you can see, the binding, the amino acid of the LISP from Pseudomonas, which is the structure we have solved, is similar in sequence with the LISP from other enteric pathogens that causes severe burden. You can see there is like greater than 96 million DALYs globally. So the main message here is that due to the sequence similarity, if you find a pharmaceutical molecule for LISP from Pseudomonas, you could potentially, they could potentially inhibit the other LISP from these other species. So the next example is the Plasmodium falciparum hexose transporter. So it's a sugar transporter. So the malaria parasite relies on sugar in the red blood cells to survive. So PFHT1 is the main sugar transporter. So it absorbs sugar from human cells into the parasite. So this is essential for the survival of the parasite. So if you inhibit this process, you could potentially kill the parasite. So we solve the structure at 3.6 angstrom resolution in the presence of glucose. So you can see the residues that are coordinating the glucose. So the binding site is also important in terms of designing inhibitors for the protein, and the structure as well is very, very important. However, there were many inhibitors for this protein, but they had only moderate anti-malaria activity. So this structure is going to facilitate the development of inhibitors to target PFHT1. So pay attention here that the resolution is 3.6 angstrom, which for structural studies might not be good enough. So another group solved the structure to 2.6 angstrom, which is higher resolution, and also they solved the structure in the presence of different inhibitors. So they published that in Cell, and they can potentially develop these inhibitors further as treatment for malaria. Another example of how structural biology provide insight into treatment of HIV. So here is the antiretroviral drug. So it binds and stabilizes the capsid hexamer. So these are 6 units. So this drug binds in the 6 units, stabilize it. So it prevents the release of viral content into the cell. So this information can open up avenue for the design of other drugs to target this HIV-1 capsid. The next example is how structural biology can help us to understand disease state. So the example here is the sickle cell anemia. Under normal circumstances, the red blood cell is biconcave in shape. It's round. And as you can see, the hemoglobin molecule, it's a tetramer, as you can see here. So the round shape enables it to flow smoothly in the blood vessel. But a point mutation in hemoglobin causes the hemoglobin to aggregate. So they form a clot. So the red blood cell becomes a sickle cell in shape, and the clot cannot flow smoothly in the blood vessel. So this simply highlights how structural biology can be utilized to understand a normal state and a disease state using the structure of both, the structure of wild type and a mutant protein. The next example is to understand how to utilize structural biology to understand mechanisms of drug resistance. So the first example here is the Plasmodium falciparum chloroquine resistant transporter. So this protein is located on the parasite surface. So when you take chloroquine to treat malaria, this protein pumps it out of the parasite. So it makes it difficult for therapeutic concentrations to be reached. So by solving the structure, scientists were able to identify the different mutations that are causing the transporter to be pumping out chloroquine. So as you can see here, these are the mutations. So this information can help us in terms of optimizing the chloroquine molecules such that the protein cannot transport the chloroquine, and this can restore the efficacy of the chloroquine molecule. The next example is to understand antibiotic resistance. So these are, they call them drug efflux proteins. So they are located in the inner membrane of bacteria. So what happens when you take in antibiotics, these pumps, they pump it out of the cell. So it makes it difficult for therapeutic concentrations to be reached. So by designing another molecule and blocking this efflux pump, you could potentially restore the therapeutic effect of the antibiotics. So the next example is how you can utilize structural biology in terms of vaccine design and optimization. So the first example here is MSP1 in the malaria parasite, which has been a vaccine candidate for many years, but there was no structure. This has hindered the progress for the design of vaccine that targets for the design of vaccine using this antigen. So the structure has been solved, and this can help in terms of identification of the antigen and epitope mapping and for rational design. So, so the next example which I'm going to highlight is on COVID, and you will see firsthand how structural biology was very crucial in the design of the COVID-19 vaccine. So if we look at the timeline for COVID-19, in December 2019, the flu-like illness was detected, and in January 2020, the causative agent was identified as SARS-CoV-2. And between January and February of 2020, this is just 1 month period, the first protein from this virus was solved. And as we speak, there are more than 1,000 structures from this virus solved and deposited in the protein data bank. So you can see here the COVID-19 spike protein, which was very important in terms of the design of a stable form of the antigen for vaccine. Then you have also the main protease, the papain-like protease, which were all utilized for drug discovery. You have other high-profile structures, which is the RNA-dependent RNA polymerase, and of course the glucocorticoid receptor, which was solved in complex with dexamethasone. So dexamethasone was shown to offer some relief to critically ill COVID-19 patients. So the next example is to understand how viruses recognize the cell before they can gain entry. So this is evident for viruses such as influenza and HIV. So on the surface of glycoprotein, you have sialic acid. And when the virus attached interacts with the sialic acid, that's when they can gain entry. Without that, they cannot. So, but how is sialic acid decorated on this surface of glycoproteins? It was unknown until in 2019, where I started working on this protein. And as you can see here, the CMP sialic acid is synthesized in the cytosol, but the glycosylation for the sialic acid to be to be decorated on the glycoprotein takes place in the Golgi. So how does it go into the Golgi, the sialic acid? It has to go through a membrane transporter, and that is the CMP sialic acid transporter. So my research determined the structure, 2.8 angstrom, and we captured the structure in complex with the substrate. And as you can see, we identified the residues that are coordinating the substrate for transport. And when you mutate these residues, you can see that some of them are very, very crucial for the function. We did the same mutation for the human version of the protein, and they all showed the same pattern. So the next example is to highlight how structural biology can be used to enhance existing medicine. So this is the case of TB. So this enzyme is the RNA polymerase enzyme, which is essential for the function of the bacteria. So this is the active site. The center here is the active site. So rifampin binds slightly outside the active site. You can see here the rifampin is the red one. It binds outside these active sites, the circle, and that causes some resistance of the bacteria because it doesn't completely inhibit the function of the protein. But then, utilizing the structure, a new molecule called pseudouridimycin was captured inside the active site, as you can see here. So it binds deep into the binding pocket of the active site of the enzyme, and this is now being used to treat drug-resistant TB. So this simply highlights how structural biology is very, very important. So despite the importance of structural biology, the African continent, basically, there are very few Africa-based researchers who actively conduct structural biology research. And when we attended the first Pan-African conference on crystallography, we realized that there were very few talks on structural biology. This is where I met my colleague Dauda, and it was evident that we have to do something to build capacity in structural biology in Africa. So this was in 2016. In 2017, we co-founded Biostruct-Africa. So this was based in Stockholm, Sweden. And the vision of Biostruct-Africa is basically to train Africa-based scientists in structural biology, to facilitate the relocations of Africans in the diaspora to establish themselves on the continent, and to also form a consortium of structural biologists working on prevalent diseases on the continent, and to establish structural biology centers at African universities. So what we want to see is that there is, we actively conduct cutting-edge structural biology research at African University, and we also want to establish regional hubs for capacity building in structural biology through Biostruct-Africa, and offer structural biology services to African researchers through collaboration. So why structural biology in Africa? So it's going to support the medicinal chemistry effort for the optimization and validation of drug leads. It's also for vaccine design and discovery, specifically epitope mapping. And it's going to create jobs for Africans and the intellectual satisfaction of knowing how genes work at the molecular level. So the structural biology pipeline, what is it all about? Basically, it's to identify a gene, and then you clone or clone and express the gene. And after you've cloned and expressed, you purify the gene and do functional assays to ensure that the gene is functional, which is very, very important. And then you can crystallize the protein if you're doing X-ray crystallography. But if you're not doing X-ray crystallography, you can do cryo-electron microscopy, which doesn't require you to crystallize the protein. Once you have your data, you can process, refine, and do the model building. So keep in mind that there are other methods of determining protein structures like NMR spectroscopy, mass spectroscopy, mass spectrometry. And also you can explore artificial intelligence. There are tools such as AlphaFold and Rosetta where you can attempt to solve the structure of your protein. So for the cloning and expression, there are different host systems that you can use to express your protein, ranging from E. coli to insect cell. So basically, the type of gene you're working on, excuse me, the type of gene you're working on determine which of this system you're using. So if you're working with a bacterial protein, you can express it in E. coli. And if you're working with a mammalian protein, it's fair to use the higher systems such as yeast, mammalian, and insect cell. And once your protein is expressed, you need to purify the protein. I'm not going to go into the details, but what you want to have is a monodisperse and homogeneous protein on side solution chromatography with a decent purity on SDS page. So once you have that, you need to crystallize it. And for that, you need a robot. And it's an expensive instrument, but you can collaborate with labs that already have this. So once you have your condition, then you can take it to your lab and proceed with the setup. So these are like chemicals used to crystallize proteins. These are the plates used. And also these are some screens. So once you crystallize your protein, you need to expose it to X-ray. And what you get is like a diffraction pattern, these black spots. And based on this, you can build an electron density map. And from there, you can build the protein model. So if your protein doesn't crystallize, you can use cryo-electron microscopy. For this, you don't need to, you just have to put the protein on a grid and collect the images. So I'm not going to the details of the method, but just have it in mind that if you cannot crystallize your protein, you can use cryo-electron microscopy. So there are the synchrotrons all over the world. They offer services to researchers from all over the world to be able to come in and do some experiments, or you can send your samples to them to collect your data, or you can ship your samples to them and remotely collect your data from wherever you are, and then you can solve your structure. And this is, we've been building capacity on this synchrotron remote access and data collection. So the first workshop we organized was in Accra, Ghana. So we structured the workshop to constitute the theoretical part, which gives introduction to structural biology and some introduction on protein expression and purification detailing. And you can see here the participants were able to crystallize the protein. You can see very happy and smiley face here. So once you crystallize the protein, you then have to ship it to the synchrotron, and then a beamline scientist will mount it on the beam, and then you can collect the data wherever you are. Otherwise, they can collect and send you the data. So here you can see that flying from Accra to Diamond Light Source, it costs costs close to I would say 800 pounds. But then you can just ship the sample, which will cost much less, and then you just stay at home and collect the data. So we were able to do this in Ghana, remotely connect to Diamond Light Source in the UK and collect data. So the feedback we got from the workshop was very, very inspiring. As you can see here, the participants recommend that we should organize the workshops annually and also try to include many participants, and also to ensure that the workshops run in a longer period of time. So after that, we planned 2 workshops. One was in Kigali, Rwanda, and another in Malawi. But due to the COVID, we were not able to carry out these workshops. Recently, we organized one at the Malaria Research and Training Center in the University of Bamako, Mali. As you can see here, we included X-ray crystallography and cryo-electron microscopy. So the participants were able to crystallize the protein and also collect data remotely and manipulate and solve the structure. So again, the feedbacks were very, very inspiring. We have another workshop planned in Cameroon. So what we realize is that there is a lot of advancement in artificial intelligence. So we want to incorporate that into our training. So we will be demonstrating AlphaFold 2, how to utilize that to solve structures, and not just that, but the data analysis and interpretation. So the participants are going to learn most of this tool, the software used for protein structural analysis, such as Chimera and PyMo. And also, we will utilize both the AlphaFold structure and an experimental structure, which we already have and know it works, to screen small molecule compound library and compare the results of both experimental and AlphaFold structure. So this will be held in Cameroon. So basically, when we launched the application, we received 100 applications, which was really, really inspiring to see. As you can see, this is the distribution. The participant came from every region in Africa is represented, and it was highly gender balanced. So unfortunately, we could only take 20. So there is need to build more capacity because we need to organize at least twice a year, so we can train more people every year. So we conducted a survey from these. So we collected the data from these 100 applicants on how structural biology can help them in their research. The rationale behind this is that most of the discovery in structural biology I have presented to you today have been led by European and US-based institutions. So it's fair to listen to Africans themselves, what they think structural biology can do for them in their research. So we analyzed the survey. As you can see, the participants came mostly from West Africa. The majority came from Cameroon, and it's not surprising to see that because it's organized in Cameroon. So it's fair that most of the participants should come from there. And also from Nigeria, a lot from Mali and Ghana, then some from Kenya, Senegal, and Botswana. So you can see they're very, very connected in terms of the disciplines, mostly biochemistry, clinical biochemistry, molecular biology, and microbiology. Some bioinformaticians. So we processed the survey and collected codes and categories in a qualitative research type format. And we obtained most of the participants say that it's going to help them in terms of vaccine discovery, drug discovery. It's going to help them in fighting antibiotic resistance. It's going to enhance their skill. It's going to deepen their understanding of protein structure and function. It's going to help them to network and provide collaboration opportunities, which is exactly what we want. It's going to help them in professional growth. It's going to help them in it's going to help in job creation. And then it's most inspiring is that they want to go back and help their institution and their local community to build this capacity. So if you see this network here, you can see how it's distributed, and you can see that there is a growing community in Africa that are really hungry for new skills, and they are interconnected in what they do and what they want to achieve. So we are very, very pleased to be at the forefront to be helping them in terms of providing them with structural biology capacity building. So that's the end of my presentation. Would like to thank the following sponsors.
Thank you very much, Dr. Nji. I would like to open up the platform for any questions that could be from the audience. Please raise your hand or drop it in the Q&A or chat boxes, and we can interact with our presenter for the day. Do you have any questions? Maybe I can start off with just asking Dr. Nji, you're doing amazing work with Biostruct-Africa. Where would my audience be? Where would they start off if they were interested in becoming structural biologists? How would they start off their journey? Or is it a classic example of the pathway that you've taken in your academics? What are the entry points for anybody who might be interested to take up structural biology in Africa?
So there are two scenarios.
Maybe you can
right?
stop sharing your slides.
Let me see. Stop. Okay, stop sharing. Is it stop now? Yeah.
Yes, it has.
Yeah.
And put on your video so that people can see you. There we go.
Yeah. So there are two scenarios. Really experienced researchers who are already working on what they're doing, and also young scientists who are like undergraduates and masters. So I'll start with the experienced ones. So the training we do now is mostly to train experienced scientists who have projects that require structural biology. And that is to provide them with the skills that they need. So basically, you can get involved in structural biology, not necessarily being an expert, but collaborating with organizations such as Biostruct to guide you through your project. But also in for those who are like undergraduates and masters, I think I look at my journey. I mostly did my bachelor in biochemistry, my master's same biochemistry, and then I did my PhD and specialized, took a project on structural biology. That was how I was able to learn the skills and techniques in structural biology. So in the future, what we're also trying to do, because most of the courses at the universities, except for South Africa, most of the courses in biochemistry don't really have like a structural biology module. So we in the past have been designing some curriculum for structural biology masters, which we think if we implement that in African universities, it's going to help to facilitate the development of structural biology at the ground roots level.
Thank you for that. I can see Sam Nji, you have your hand up. Okay. Please unmute and go ahead. Sam, can you hear us? Samson Nji, can you hear us?
Yeah, now I can hear you. I had not been enabled to unmute. I think
Sorry
I've been
about that.
muted. Okay.
Yeah.
First, Emmanuel, thank you very much. A really interesting presentation. I had capacity building at the program, so your language is a language I like hearing. And I think in terms of just building the general structural biology skills, it's a really good vision and goal. My question is also then going forward, because, you know, as you showed, structural biology is quite expensive. And so how do you think we can move from now building just the individual skills, which is great and I'm very supportive of, to then finding resources for putting together centers of excellence, or even a center of excellence in Africa for structural biology that then takes the skills that you will develop into actual practical application?
Yeah, this is a very, very good question. I'm really delighted to hear you mention the African Center of Excellence for Structural Biology, because that's something that has been going on in my mind. Firstly, I totally agree with you that the facilities for structural biology, the main facilities, are very, very expensive. But they especially the synchrotrons. So they created these facilities for, these are governments coming together to build this facility because it's such an expensive facilities. So when I was in Stockholm, I was still flying to the UK to collect data. So people fly from different countries also in Europe to come and collect data here. So it's not that we are limited in that sense. Because if we were to build a synchrotron in Africa, I have checked most of the time, it would, which they are advocating for, to have our own synchrotron in Africa. Most of the times, what we notice in this workshop we're organizing, like booking the tickets within Africa, is much expensive than booking from Africa to Europe. So it means flying from Kenya to maybe Ghana to go and collect data at the synchrotron will be it would be cost effective to just fly to Europe to collect the data there. But that's notwithstanding, they've made the synchrotron such that you can do this remote data collection. where you can send your samples and then you collect your data back at home. So it was designed for this purpose to enable those institutions who don't have facilities to be able to also benefit from these instruments. And also they have collaborations as well, where, for instance, I am now in the Diamond Lightsource in the UK. They have collaborations where you can fly and do your experiment. They call it rapid access. So you can fly and do your experiment. So in that sense, they are also like building capacity for Africa-based researchers. So if you go there, you're learning the technique, and then hopefully you can take it back to your continent. So I think collaboration will be key. The remote access will be key as well. And then in terms of the building capacity, we are now going also to focus on the computational side of structural biology, because it's getting really, really interesting with the artificial intelligence, AlphaFold, Rosetta, which is helping a lot of structural biology. So this artificial intelligence doesn't eliminate structural biology, it helps it, so it facilitates it. For instance, the structure we solve, we use the AlphaFold structure as a model to solve the experimental structure. So even in terms of vaccine design, you can pop your sequence into AlphaFold to see your antigen. It can guide you, give you a guide on what part of the antigen you can focus on, which part might be disabled, what you need to, maybe you need to stabilize it more. So the computational side, in addition to the experimental structural biology, we have to do them in parallel. And hopefully that is going to help us. I don't know if I answer your question correctly.
Yeah, you did. Thanks. Yeah.
No worries.
Thank you for the question, Sam. Do we have any questions from the audience? I have a question. With Africa, where it is the lessons learned during COVID, and the current fear of pandemic X, where exactly does structural biology fall in the development of vaccines? Is it something that Africa already has a capacity to do? Or are we very far off, and what would be the priority? So those are two questions. Where are we as a continent in terms of preparation, being prepared for pandemic X? And two, what would those priorities be? Which particular diseases currently are in the pipeline that need us to pay attention to?
Yes, thank you for your question. This is really, it's really a serious question. Yeah. So in terms of pandemic, so this would be like a global thing, right? So so if that happens, then I think structural biology, then people unite in terms of doing stuff to be able to solve structures of proteins, to see antigens and so on for the vaccine design. That was the same situation in the COVID time. So during the COVID time, the synchrotrons were open only for research that is related to COVID. So if you're working on malaria or whatever, no, you don't allow access. So they give priority to those people. That's why they were able to speed up the process. Because we don't have those facilities in Africa, so we definitely rely on foreign institutions to get data to get structural biology data for vaccine design. But I think the way it's going at the moment in terms of artificial intelligence, it could prove useful at some point in the future.
Yeah.
But even at this point in time, there is still a reservation as to what you can get from artificial intelligence, even from those who designed everything.
Yeah.
And then in terms of priorities for diseases in Africa, I'm not really like familiar with the other emerging diseases in Africa, but I know there's lots of I know malaria kills a lot. I'm not sure if it's the #1 killer in Africa. Do you have any idea on that? I think 500,000 people die every year from malaria. But the burden, also you look at the burden caused by the disease as well. So it's something to pay attention. And also other emerging viruses that are coming up, right? In things like Ebola, which is very deadly. Have to also pay attention to those. Yeah, I mean, that's just what I can think of at the moment.
Thank you for that. I can see Cleopas, kindly ask your question. Cleopas Elisha, can you hear us? Okay. I don't know whether he's being able to hear us. He has his hand up for a question.
Maybe he can, if he can speak, he can type it in the chat.
Type it out, yes. So as you wait for him to...
Yeah, someone made a very important comment here. So is it Samson? So he said, even our power supply is too variable for such high precision equipment. Yeah. No, I totally agree. We really have to. So there was this, it's called, there's a STARTS program. It was funded by UK Research, is it UK Research Council? It was like 4 million British pounds to develop synchrotron science and energy in Africa. But then it's that can't really help in a lot of countries, you know. So the research was awarded to South Africa. But then lots of other countries, most of the institutions have like a backup generator to even run like protein purification smoothly. So it's really a big problem that Samson is highlighting here. Yeah.
Somebody has sent a Q&A on, since Biostruct is doing the training, do you also do the placement once they get the skills that are needed? And where would those collaborating partners be placed?
So we recently got a grant from Swedish Research Council and former Sweden. And this grant was to train, to organize workshops, and also for researchers from Africa to go like exchange visit in Sweden. Also, I'm based in the UK here, in the Diamond Light Source. So they have like rapid access, where if some of the participants we train wants to do some experiment, we can link them up with these institutions. Also, what we really want to do is to form a network. So we don't want to train people and cut them loose. No. That's why we're very, very stringent in what their initial research are. So we understand that it's something that's likely going to go in that direction during the selection process. So we form a consortium, which we'll have like online mentoring. So we organize something like webinar, like this webinar you're organizing, something very, very important. And it's something we also have to do to help them and keep them informed with the structural biology landscape. Yeah.
Okay. I like the idea of the consortium, so you're able to also share the skills and knowledge and opportunities that come up.
Exactly.
Yeah.
And we can collaborate, write papers together, publish together.
Mm.
So everybody win-win.
Great.
Yeah.
Cleophas, I'm not sure whether you can still hear us. If you can, please ask a question, or you can drop it in the chat. Not sure whether he can hear us. Any other question from the audience? Okay, so from me to you, it's just to say thank you very much for everyone who has attended this webinar. This was quite informative, and I believe we have walked away with new knowledge that we did not start off with. And for me, it's just to say thank you very much to Dr. Emmanuel Nji for making the time and for imparting his knowledge. I hope you all have a good day, and I'm looking forward to more engagement with you in upcoming webinars. Have a good evening and have a productive day. Thank you very much. Goodbye.
Yeah, thank you.